welcome everyone and to this distinguished lecture series. And I found this room is hiding. It's a little bit hard to find. At a room door it says 1103. Because I saw my colleagues who asked for the seminar, they haven't shown up yet. But I think the time is limited. We're going to start now. Yeah, it's our great honor today. We have Professor Howard Stone from Princeton coming to give us this distinguished talk. And Professor Howard Stone got all his degrees from UC Davis and went to the Caltech for his PhD and did a short-term postdoc in Cambridge before he joined Harvard for 20 years. And in Harvard, he was working at the School of Engineering and Applied Sciences. He won 2 teaching awards. That teaching awards is the only 2 awards given to faculty members in Harvard. And in 2009, he moved on to Princeton. And he's Donald Dixon and Elizabeth Dixon professor. And Professor Howard Stone, and he said, okay, make the introduction very simple. But
you can stop now.
I still want to say a few words. So yeah, the Professor Howard Stone, won so many awards, but I'll just say a few of them. And he was elected to the National Academy of Engineering in 2009, American Academy of Arts and Sciences in 2011, and the National Academy of Sciences in 2014. So without further ado, I'll give the room to Professor Howard Stone.
Okay. Can you hear me? Okay, so first of all, thank you for the invitation. I'm going to talk to you about some work my group has done that involves 2 rather distinct subjects. What they have in common is that they involve flow, because I study fluid mechanics problems, and they involve very simple geometries, so simple that at least in one case you're going to be sure that this is in all textbooks, and I'm going to try to convince you it's not. I should start with an apology. We're in the Institute for Quantum Computing. There's nothing quantum about this talk. There's certainly, there is some simulations at the end that I need to try and rationalize something, but there's no computing in the sense it's normally done. And I'm visiting the Institute for Nanotechnology, and arguably you're going to have to look hard to see if there's anything nano in the talk. But I hope that I can tell you something that you're going to find maybe surprising, maybe interesting, and we'll see as we go. The work I've done has been done by a very talented group of people, my students and collaborators. My group at any given time is about 1/3 visitors. Cody Chen in the audience just spent 6 months with our group. And I get to collaborate with a bunch of people in molecular biology. Professors Emmer Gittai, Bonnie Bassler, Ned Wingreen are all molecular biology professors that I'll tell you a little about our work today. And a variety of other kind of short-term visitors, long-term visitors, graduate students, postdocs, and the children in the presentation, I suppose, have nothing to do with the talk other than we're all human. Okay, so here's the outline for the talk. I'm going to start with a couple slides that just try to convince you fluid mechanics is interesting as a way to think about the world. And I'm going to have some short stories about bacteria and biofilms, and what is special about them is I'm going to tell you about bacteria that live on surfaces, and in the fluid mechanical perspective, since there are fluid mechanical people in the audience, the Reynolds number in this first part of the talk is a small number. I'll tell you about 2 different things that happen when you put flow over bacteria on surfaces. You are allowed to express skepticism that anything interesting should happen, since the bacteria are on the surface. The velocity at the surface is very low. What possibly interesting could happen if you have bacteria on surfaces? And one reason I say that is the most common problem in fluid mechanics involving bacteria are swimming bacteria. There's a large literature. It's a field in mechanics now called active matter. The second part of the problem is a high Reynolds number flow problem. Now, I started life doing low Reynolds number hydrodynamics. So with apologies to some people that do turbulence in the audience, high Reynolds numbers for me means anywhere from 100 to 1,000. I'm going to show you flow at a T junction, which is a very simple shape. You've seen it in textbooks, and I'm going to try to convince you that the textbooks and all the researchers missed the most interesting thing associated with the flow. And then I'll try and convince you it's also important, possibly. Okay, so first, fluid mechanics, why is it interesting? This shorebird lives in shallow water. When it gets hungry, it does the following. Anybody know why? Oh, I should say it lives in shallow water. It doesn't like to get its head wet. And so it does this little routine so it can, it's pecking, it's essentially eating. So the shorebird is making, because it lives in shallow water, it's making a little vortex. The vortex brings up organic matter at the bottom, floats to the surface, and the shorebird can eat without getting its head too wet. Amusing? Okay, so I was at a conference, an APS conference. There were 1,000 people in the audience. A former student of mine was giving a talk, and he showed this video. At the end of the talk, there were no questions. I hate it when there are no questions. So I stood up at the end of the talk and said, does the fact that the shorebird knows to do this stirring of the fluid mean the shorebird knows more about fluid mechanics than we do? And my former graduate student said in front of 1,000 people, I can't speak for everyone in the audience, but I can assure you the shorebird knows more fluid mechanics than my PhD advisor. Okay, so that was item one. Item two, leading cause of death in the Western world associated with eating red meat. Your heart is a pump, so it's a mechanics problem. When it fails, you die. And the basic feature is a modest effect associated with the radius of a channel getting clogged by fat, or if you like going to France, too much cheese. But it's a reduction in radius. And this poses a fluid mechanics question that you can pose the following way. If I give you a pressure drop in a pipe of a certain length, and the pipe has a certain radius, and the fluid has a certain viscosity, please tell me the relationship between the pressure drop and the volumetric flow rate through the pipe. The standard way, by the way, textbooks answer this question is by using calculus, writing down an equation, and then integrating. But as I've stated it here, you can answer everything by dimensional arguments alone, provided you recognize the dimensions of viscosity, which is standard in a fluid mechanics class, and the fact that you don't need the pressure drop and length independently. All that matters is the pressure drop per unit length of the pipe. If you make that statement, then dimensional arguments lead to the following formula, which is well known as the Poiseuille formula, or as a French-Canadian student once insisted to me after I had mispronounced it, said the Poiseuille formula. The volumetric flow rate is proportional to pressure drop per unit length, inversely related to viscosity, but it depends on the fourth power of radius. And for those of you in microfluidics, you know this is a large effect, not a small effect. And so this geometric effect says that unlike many engineering problems where small causes produce small effects, this small cause produces a significant effect. And in fact, it raises the question that if you have a 10% reduction in flow rate, 10% reduction in radius, what's the reduction in flow rate? Because of the power 4, it's effectively a 40% reduction in flow rate. So these geometric effects can be large, and I'm going to show you one in the middle of the talk that isn't profound, but wasn't pointed out in the literature in the subject of bacterial clogging. Okay, so these are just two little things about fluid mechanics that I find interesting. And just to protect myself, of course, dimensional analysis won't give you the pi over 8, but it gives you everything else. Okay, so now some short stories. First, I want to tell you a little about bacteria, biofilms, and fluid mechanics. I should say that each of the little stories I'm not going to tell you has the following feature. I'm going to try and tell you an idea. I'm going to show you an observation. The observation, I think, in every case is going to surprise you. I'll try and give you the idea that there's some implications of it. I can't, because many of the things we do are lab-based, I can't claim that they occur in the real world, but everything about the things I'm going to show you are completely generic in the lab. And in most of the problems, we've done different kinds of mathematical modeling or numerical simulations. I'm going to suppress that. If you have questions, I'm happy to answer them. OK, so As I said, the most common problem in the fluid mechanics area involving bacteria involves swimming microorganisms. What you should know is most places you find bacteria, there actually is flow. You find it in the vasculature of plants and animals. You find it in all manners of food processing, in the function of all your organs. And if you end up in a hospital, all kinds of medical treatment devices involve flow through small devices, and they might be contaminated by bacteria. Many common bacteria are rod-shaped. The one I'm going to tell you about today, Pseudomonas aeruginosa, typically has a single flagella. It's a rod-shaped bacteria that's roughly about 1 micron to 2 microns in length. What's interesting to me is that there are many studies of bacteria, and there are many studies of fluid mechanics. There are, in fact, very few studies of bacteria on surfaces when there is flow. When we started this work, there were a handful of people, very nice studies about this problem, but it was relatively sparse in terms of investigation. So I'm going to tell you about the combination today of bacteria, in particular bacteria on surfaces. When they develop a lot, they're called a biofilm and flow. Okay, so here's a question you're going to have to be prepared to answer in about 4 slides. I'm going to show you a network. This is a picture of a vasculature of an animal. In the lab, I'm going to show you a network that has some of the features of that microfluidic network. At the end, I'll also show you a plant. And the mechanics question we're going to ask is, the arrows indicate the flow direction. All the flow will go left to right. We're going to inoculate bacteria at one location in the pipe. And you're asked the question, where does the bacteria end up in this network? The reason this is an important question, of course, is it takes one bacteria to kill you because they will multiply. And if you ask any person that does transport phenomena, and I will go out on a limb to say any person that does computer modeling, their computer model will tell you the bacteria will end up following all the flow lines and will end up in the upper branch. The question is, where does the bacteria really end up? And so in order to introduce that idea, I'm going to tell you the following story. This bacteria, Pseudomonas aeruginosa, is a very common pathogen. If you have a compromised immune system and you're in a hospital, it will kill you. It's this rod-shaped bacteria with a flagella. When the bacteria sticks to the surface, it loses its flagella. Its flagella is for swimming. It has other appendages along its body that are called pili. All the pili help you adhere to help the bacteria adhere to the surface. But these long appendages in front, these so-called type 4 pili, in Pseudomonas aeruginosa are motorized. They're via polymerization process can shorten. In the process of shortening, they apply forces that pull the bacteria along the surface. And that, in the language of molecular biology, is called twitching motility. The common idea about this twitching motility is it lets the bacteria randomly search its surface, maybe for food or whatever thing, but it lets you kind of do random motions. That's the suggestion. What my graduate student Yi Shen discovered was that when Pseudomonas aeruginosa is exposed to flow, and we use standard microfluidic methods to study this, something else happens. And to make the experiment clear, and there are many experts in the audience who know much more than I about microfluidics, but the flow is from top to bottom. Think of the boundary being a zero velocity boundary. There's certainly a stress that the fluid will put on the bacteria. And now you ask, where do the bacteria go? And in the presence of this flow, some fraction of bacteria are going which way?
Upstream.
Yeah. So I'm going to use the word discovered. It turns out it's an overstatement. What we discovered, it's just easier to say, it sounds more impressive, is that in the presence of flow and this twitching motility, some fraction of the bacteria go opposite the flow direction. They go upstream. Okay. And I'm going to tell you now a little about that, how it happens, why it happens, and a consequence of that. Okay, this was one of the first things we did. I'd like to say we discovered it. It turns out when we were about to submit our paper, we found in the literature an obscure but very beautiful paper by a group of people that study plant biology who had identified a pathogen that lives on a leaf, starts on a leaf, and ends up inside the plant. And they hypothesized that since the plant is pumping water upward, that the bacteria was somehow working its way opposite the flow of water. So there was already a hint in the literature in this paper, and we've kind of analyzed this in great detail. So how does this happen? I told you that this bacteria has appendages that are motorized. I showed you that movie. And these appendages help you stick to the surface. So when a rod-shaped bacteria sticks to the surface, if it experiences a shear flow, it experiences a stress. The stress is just going to flip the bacteria. And it doesn't really matter now how the bacteria falls on the surface. No matter what orientation it falls on, it's going to have some component of its direction of motility directed upstream. And so the consequence of the rod shape means you flip. The consequence of the motility means you now walk upstream. And the evidence for that is going to be shown by the movie at the bottom that a man named Alex Persach suggested to us. We did this movie where we put particles in the flow to show you that the velocity is going top to bottom. Halfway through this experiment, we're going to flip the flow direction. This bacteria, in fact, is about to divide. You can see it's 2 cells almost. And when you change the flow direction, the bacteria flips because it's polar localized at the attachment point. So this is indirect evidence for exactly how this bacteria is able to migrate upstream. And what is the consequence of that? So a consequence of that is this experiment. So now you ask, if you have a bacteria that lives on surfaces but is motile, and you say, well, it's like anything else, it's like colloids, you solve convective diffusion equations. That just tells you that things follow the flow. But here you have an object, because it's living, is capable of moving opposite the flow direction. And you have a network that has junctions that change where you go. So we're going to inoculate the flow where the laser pointer is pointing. The experiment, in fact, uses 2 types of bacteria. It's the advantage of being an engineer that doesn't know much biology but has many colleagues in molecular biology who are both nice people and world-class scientists. So we test wild-type bacteria which have motility, and a mutant called PIL2U, which has the appendage, but it's not motorized. It can't do this pulling, so it's a stationary bacteria. And the consequence of this is now when you do this kind of flow experiment, the non-motile bacteria is confined to the upper channel. It actually forms a biofilm-like structure. The black bacteria, which is wild type, some fraction of them can walk upstream. When they lose connection, when they lose contact with the surface, they end up in the lower channel. And the consequence of that is you end up with bacteria now in a network that you would not otherwise suspect if you weren't aware of this local feature. Okay? So that's short story 1. So you end up with, in this bottom channel, bacteria. And we've been doing a little work over the last 1.5 years to try to make an effectively convective diffusion model of a system that's a network with both motile objects capable of upstream migration and non-motile objects, just to show the effect. Okay. Anyway, so that's short story 1. Upstream migration in flow for trying to predict the possibility of infection of surface motile bacteria. Okay. The way we then applied it in our paper is we took a plant. The plant is driving flow from the stem to the right side. There's a place where we will inoculate an infection, and we do the same kind of experiment. The green bacteria is motile, the red bacteria is non-motile, and over the period of a week, the green motile bacteria can find can migrate all the way to the kind of the source of the flow to the leaf, while the red bacteria is confined to where you place it in the system. So at least that's an artificial experiment with a living system that shows the same kind of upstream migration, with here a flow that's driven by the plant. Okay, so that's short story 1. We almost had nano. I mean, if you really insist, you know, the appendages are nano objects. Well, I'm not going to apologize. You invited me, so you get what you get. Okay. Okay, so the next thing I want to move from individual bacteria, I want to move to collectives of bacteria. Collectives of bacteria are called biofilms. And this project has grown over many years with a large group of people. And biofilm is a collection of polymer and cells, and there's bacteria on surfaces. So if you have children that don't brush their teeth in the morning, the gunk that forms on the teeth is a biofilm. If you're out in the wild and you end up with kind of slimy surfaces, that's a biofilm. If you look at wounds or pus, there are biofilms there. So biofilms are lots of places. Again, there's some combination of cells producing a polymeric material that is both adhesive and protects the bacteria. We generally think about these as bad. If you're a material scientist, there are lots of efforts around the world to try and design surfaces to minimize the growth of biofilms. But biofilms can be good. They're used in water treatment. You have inside your mouth something like hundreds of species of bacteria that help keep you healthy, et cetera. All right, so these are very commonplace. And it's been now known for 30 or 40 years that the most common bacteria are in biofilms. They live on surfaces. They're not planktonic or free-swimming bacteria. Now, this goes back a number of years, but we started to do flow experiments with bacteria, a little like the ones I showed you. I can't exactly remember why we began with this experiment, but it went something like this. We were doing microfluidics. We were asked to look at how surfaces respond to flow when there are bacteria around. So we did so-called low Reynolds number experiments. Oh, I'm going to color lots of things green now because the bacteria we're going to use, the Pseudomonas aeruginosa, has a green fluorescent protein in it. So where you see green, they're bacteria. We made channels that had these kind of funny shapes with bends in them. It was partly accidental. I'm going to try to show you in a second that it's important. We use this bacteria I already told you about. We do experiments. We started by doing 10-hour experiments. Turns out that's important. We ended up doing 100-hour experiments. Green is fluorescently lit cells. We have a whole study on mutants I'm not going to tell you about. Just to defend myself a little, since the most common channel is a straight channel, why might bends matter? Everyone in the audience is sitting with your elbows bent. Both in this part of the talk and the last part of the talk, I'm going to claim that that's an important thing to study, and it's not my fault that most researchers and most textbooks don't pay a lot of attention to it. And the second thing is we have to do long time experiments. You know it's very common to get criticized in the literature and proposals or whatever. You live 7 days a week, 24 hours a day, a long time. It turns out in this problem there's a time scale that we don't completely understand that's associated with rearrangements of a material. In rheology, you call that a time constant of a material. It tends to be somewhere from a few hours to many tens of hours. We got lucky in our first few experiments that we were longer than the time scale, so we saw something interesting. And you need a long time experiment to see that in these systems. So two things happened that were accidental, both shape and time, that turned out to be important. So here's the first part of the story. If the channel is straight, we never see anything interesting. The bacteria, we grow them on the surface. They're on the surface all the time. They just slowly get bigger. The biofilm just slowly gets thicker in time. Okay. To make that clear, if this was the inside of a pipe, the biofilm would be on all the surfaces. Okay? And it would just accumulate in time. When the channel has a bend in it, and as far as we can tell, it doesn't matter what the bend looks like, any kind of curve. When the channel has a curve in it, something else happens, and that's shown here. We're going to take, in the middle of the experiment, we're going to take a confocal microscope, and we're going to scan from the bottom of the channel where you have a biofilm to the top of the channel where you'll also find a biofilm. And if you do that some number of hours after you start the experiment, when you leave the surface, you lose the biofilm. This image, it turns out, I'm going to show you, is exactly in the middle of the channel, and when you leave the middle of the channel, you find the biofilm in the other wall. Okay, so what this experiment shows, which as far as we can tell, no one in this literature had ever noticed when they had been studying how soft materials clog, is that in the middle of the channel there's a string of biofilm. Okay, so this is now the second part of my talk. I'm going to tell you about something floating in the middle of the channel. Okay, I've watched this movie hundreds, if not thousands of times. I can tell you the following story, which professors and graduate students both might appreciate for different reasons. When my team came, when my group came and showed me this experiment, and they said, look at this. I said, it can't happen. It's impossible. And I said, why it's impossible is I've studied low Reynolds number hydrodynamics for 20 years, and I know a lot about low Reynolds number hydrodynamics, and there's nothing interesting about low Reynolds number hydrodynamics in the system, especially in the middle of a channel. And they simply looked at me and said, it happens in every experiment we do. And I'm now going to try and tell you the story behind this. And I should say also, it's quite generic, I think, what I'm saying. So the reason for that statement is we then started to do channels where we took arbitrarily shaped channels, some with sharp corners, some with round corners, some with short distance between corners, some with long distance between corners. We, again, I forgot to say this, we first prepare a biofilm, and then we start a flow. And whenever we see this string of material, which had been noticed in a few places and called a streamer, so we'll call it a biofilm streamer, if you see it in one place in the channel, and you look many other places in the channel, you also see it. So it's not as if, or at least we have lots of evidence that says it's not true. It's not as if you pull a string out of your experiment and extend it along the channel. If you see it at a corner, you see it at many corners, and wherever you look in the channel, you see it at corners. Okay? It seems to be a corner effect, an effect associated with changes of direction. Okay? So we refer to it as a biofilm streamer. I've shown you a video that says it's sort of in the middle of the channel. And so if you do a three-dimensional confocal reconstruction of this structure, this is kind of smoothed out. There's a biofilm at the bottom. There's a biofilm cut off at the top. And floating down the middle is a string of biofilm. Other images of this show us, make it look a lot more like many filaments of biofilm, but all localized near the middle. You're allowed to say what's special about the middle. I'll try and tell you what we think is associated with that. Now, let me say why this picture is important if you go into the literature on engineering of systems or how things clog. The standard picture of how things clog, which is in engineering literature, physics literature, applied math literature, biology literature, is biofilms grow on surfaces. Flow delivers nutrients to surfaces. Things grow on surfaces. And clogging happens because things start on the surface and they grow inward. That's in every model. Well, if there is a model, the model has that feature. What I'm going to show you is that, at least in our experiments, sometime after you start the experiment, you put a string in the middle of the channel. And the string grows from the middle outward. And our experiments always clog from the outside. I'm sorry. Our experiments always clog from the inside out, not from the outside in. They're completely opposite the way every model in the literature tells you clogging happens, because every model in the literature makes a math statement, which is, oh, it looks like a reaction diffusion problem. Ours looks like the picture on the slide. Okay, so it happens with this bacteria, Pseudomonas aeruginosa. It happens with most bacteria we've tried. So we've done different kinds of bacteria, and we always see the same kind of structure associated with corners and biofilms, these so-called biofilm streamers. Okay, it seems to be a corner effect. I'm not going to, so here's a slide that suggests it's a corner effect. In the upper right is a fluid mechanical calculation we've done. I'll tell you about in a second. The experiment we tried to do was, we tried to set up an experiment a certain way. We tried to, say, run it. We decided 20 hours. And then for channels that were nearly straight or slightly more bent or more curved and increasing curvature, we tried to change the curvature at the corner. We took a picture at the identical times, and we showed experimentally that as you change the curvature, you get more biofilm at the corner for the same amount of time. So we correlated the formation of the so-called streamer structure, the accumulation of it, with the radius of curvature at the corner. The upper right is a single-phase fluid mechanical calculation where we used asymptotic methods for this problem to show that the strength of the vorticity, or what's called the axial vorticity at a corner, increases with the radius of curvature in the corner. And the axial vorticity is associated with the idea of not a axial flow, but a flow with swirl in it, which a fluid mechanics person would call the secondary flow in the neighborhood of the corner. So we use theory and experiments to suggest that it's what's called the secondary flow at a corner is associated, let me skip ahead, and then I'll show you a movie. The secondary flow at the corner in a low Reynolds number system is associated with these structures being formed. The secondary flow in the corner is just, you know how to sketch flow along a pipe, so now you have to say the flow is actually swirling. The theory of low Reynolds number hydrodynamics tells you that around a symmetric corner, there's a plane of symmetry, the flow has to change sign, if you like. The secondary flow has to change sign in front of the corner and downstream of the corner. So upstream of the corner, it turns out the secondary flow is going away from the middle of the channel. Downstream of the corner, the secondary flow is going towards the middle of the channel. And we've suggested, though we can't prove, that it's the nature of the secondary flow that's accumulating things into the middle of the channel that's causing this kind of effect. Now, how these things get formed is, I'm pretty sure, is a rheology question for those of you that study material science or rheology. This experiment, the flow goes from the bottom to the top. There's a biofilm on the surface, and sometime after starting the experiment, you extrude a string, which is, we think, a polymeric filament. It tries to follow streamlines and can't. It then catches everything in the flow, which is bacteria and small stuff, and you start to form a filament that goes from one corner to another corner. So the way we model this is we combined elasticity and fluid mechanics. A filament in a flow, it turns out, can't follow a streamline. It behaves, we modeled it as Euler's elastica or something like that. And so we tried to understand what do soft filaments do in a fluid flow. And so this movie gives you an idea that there's something about the nature of the softness of the material, which is a rheology question, not a biology question, that's leading to the formation of these structures. Okay, so now, how do systems clog? So here's why a small amount of biofilm in this experiment matters not a little, but a lot. Oh, and I should say, I'm having a senior moment. What was I about to say? Oh, yeah. I need to convince you the following thing. Or at least I want to suggest the following thing. Any kind of porous media is going to have these features. It's going to have small dimensions, sharp corners, round corners. It's going to have random stuff. So you might, my claim is if you see this in this kind of experiment, then any kind of porous system you see is likely to have these features. Low Reynolds number flows, change in flow direction, et cetera. Okay, so here's then a math question you can answer. Suppose I give you flow in a pipe. Pipe has radius r0. And now you reduce the radius of the pipe epsilon, right? How does the flow rate change? Well, we saw that at the beginning of the talk. There's the Poisson formula with the radius to the 4th power. So when you reduce the radius epsilon, you reduce the flow rate 4 epsilon. So the textbook problem is that when you have a biofilm present and you've reduced the radius a little, the flow rate goes down a little. I'm going to put epsilon equal 0.01 into this formula in a second. It's pretty boring, right? Okay, but here's a textbook problem that's in fluid mechanics textbooks. It never made it into the literature on biofilm clogging or bacterial clogging, which is that our experiment shows that sometime after you start the experiment, the biofilm isn't just on the surface. It's in the middle of the channel as an elastic material. So now you solve a flow in a pipe with a pipe at the center, and you make the radius of a pipe the same amount of biofilm that's in the top. And if you do that, you've now put a no-slip boundary condition, effectively, in the middle of the pipe where the flow is fastest. And now the change in flow rate is given asymptotically when epsilon is small by the formula Q biofilm divided by the presence in the biofilm compared to the clean channel is 1 plus 2 over log epsilon. And if you put epsilon 0.01, 1% biofilm, it doesn't do anything to the top. It reduces the flow rate in the bottom one 50%. So here's a geometric effect that's catastrophic to you. It's not a little effect. Because it's a 3-dimensional effect, it completely changes the pressure drop flow rate relation. To give you evidence that this is roughly what happens, we did experiments where in most microfluidic experiments, well, in my lab, we do flow rate control. Here, you guys are smart, so you do pressure control. We did pressure control with just a little hydrostatic head. We imaged growth of the biofilm during the pressure-driven flow, and we kept track of how much material came out. I didn't have a lot of money, so we couldn't buy fluid pumps, so we just did things this way. And we measured mass flow rate as a function of time. And I thought things were going to kind of be boring, but they look like this. For most of the experiment, nothing happens. So for 50 hours in this experiment, nothing really happened. The flow rate went down a little. Why? There's a little biofilm on the surface. As soon as the biofilm streamer forms, it rapidly grows in time, and the flow rate goes to 0 over about 2 hours. So here's evidence for this, if you like, clogging from the inside out. And this idea that to understand this problem, there are at least 2 time scales. There's a long time scale until the streamer forms, and then there's a short time scale associated with the rapid formation of the streamer. And we've tried to study some part of this, and we've tried to see what biocides do to it, et cetera. But I'll just stop with that, and I'll just show you one more thing. Why might this be real? We did experiments with a number of real systems. Here's a water filter. The water filter is made up of a metal mesh. It's sold by major companies. It's very common. The picture of biofilms that you'll find people talk about is biofilms are on surfaces. In our experiment, once you form a biofilm on the surface, you also get these streamer-like elements forming a mesh-like network over everything. So these streamer-like elements are found in other places, not just in this little laboratory experiment we did. OK? So that's my second part. So I'm now going to go to the third part. But before the third part, I have a little short story. So, and the short story is about this man, Hardin McConnell, who was one of America's great physical chemists in the 20th century. And I had the good fortune to work with him a short time. He studied physical chemistry problems, but he started to work on some fluid mechanics problems. And I knew some people in his research group. He was at Stanford. And one day, one of his graduate students sent me an email that said Hardin left the group meeting furious. And he was muttering the following phrase as he walked out of the meeting. He was a physical chemist. Biology was invented by God. Physical chemistry was invented by nice people. Fluid mechanics was invented by the devil. OK, so I've always liked that phrase. OK. All right, so the last thing I want to tell you about is another simple geometry. It again has a bend in it, so I'm going to refer to your elbows. If you don't think this is important, then keep your arms straight for the rest of your life. I'm going to tell you about a 2-phase flow to T junction, but some of the things I'm going to tell you are single-phase flow results. Large group of people have contributed to this. The Reynolds number here is fairly large, so the explanation I'm going to end up giving you is different because it's a high Reynolds number, not a low Reynolds number flow problem. And I think this is interesting because this T-shape I'm going to tell you is just common. It's lots of places. How common? Well, you find it in the lab, a T-shape. You find it in all piping systems where you're distributing fluids. And if you look in any organ system, this is some of your physiology. You'll find these T-shaped junctions. Now, there are studies of this. I'm not claiming we're the first. There are lots of studies of this kind of problem. You find it in all, power plants. People study this in industry. Academics study it for numerical reasons and fluid mechanical reasons. There is one thing special I'm going to tell you about, which is in every case I'm going to tell you about, the flow is going to come from the top of the T and branch symmetrically to the side. You can do this other ways. You can come from the side and go out the other two. But in every case here, we're going to come from the top and branch. Right, the Reynolds number is big. The Reynolds number is defined just like you do. I've given this talk a bunch of times. I've never been able to get a grant on this for 4 years. I'm going to now go out on a limb. I'm going to claim that I'm going to show you something new in fluid mechanics. It's not in the research literature, except for our papers. It's not in textbooks. I'm going to show you why it should be. I'm going to claim also a new kind of instability in the fluid mechanics literature that no one pointed out. And I'll go out on a limb to say there are lots of numerical studies of this, but all the numerics people missed it. And they missed it because they never asked a good question. Oh, I'm on tape, aren't I? Bad, bad, bad. They didn't really make a careful plot of what happened as the Reynolds number systematically increased in this configuration. And if they had, they would have discovered an unusual effect. That's what I'm now going to tell you. So here's the experiment we do. Flow in a T junction. in at the top, out at the sides. Reynolds number is ordinary. We were first looking at, oh, I should say, you know, you go to a professor and you say, sketch the flow. We were talking about this morning, Jean-Pierre, right? And if you were to sketch the flow, this is what I did. I said, well, it sort of looks like that. Yeah, how could it not? Flow comes in, it goes like that. If I ask you, there's a stagnation point at the boundary, and if I ask you, if I put objects in the flow, is a stagnation point a stable point or an unstable point? What might you say? Put objects come in, they're going to go away. So because of that, you might say, from a nonlinear dynamics point of view, if you were a person, you'd say, well, it must be an unstable point. So if you put rigid particles in the flow, these are glass particles, so they're about 3 times as dense as water. They come in at the top. There are some complicated trajectories, but they all leave. I should say we were doing this because we were trying to understand the speed that particles impact the wall to understand damage to a wall. There's a short story behind that. But this is what a typical experiment looks like when the Reynolds number is a few hundred and you slow down the video. Except one day, bubbles got stuck in the flow. So gas bubbles, a standard 2-phase flow. And when gas bubbles get stuck in the flow, and you take a high-speed video of the flow, you see what's on the video screen. And it doesn't matter, by the way, what the orientation is of the pipe. This is the effect that happens. Things accumulate there. They don't leave. Kind of see that? OK, so this is the story that I want to suggest. There's something interesting about this geometry. This, as far as we can tell, wasn't in the 2-phase flow literature and it wasn't in the single-phase flow literature. Everyone see that? I now want to just spend 10 minutes and tell you about this kind of unusual observation that I think is rather, well, might be interesting because it's such a simple geometry. And let me just emphasize, bubbles are important in this case. What really is important is density difference. But as far as we can tell, gravity doesn't play any role in this. It doesn't matter what the orientation of the pipe is. I should say in this experiment, the pipe's about 1 millimeter in width, square cross-section. The bubbles are a few hundred microns. We typically add some surfactant. It's natural surfactants present that sometimes keeps the bubbles from coalescing. OK, but things accumulate. So now I get to try and explain where this comes from. I should say, and I told someone the story this morning, we had this for 2 years. We had all these observations. We knew it happened. We had categorized it. We had no idea why. And the reason we had no, well, one reason we had no idea why was I was making a blunder. And the blunder was the slide I just showed you about sketching streamlines. I kept sketching this, and I said, how does this flow ever trap anything? And this, as far as we can tell, this sketch, which all professors, I think, would typically make if they weren't thinking clearly, it has a flaw in it. This is a 2-dimensional sketch. And what I'm now going to tell you is this flow is fundamentally 3-dimensional, and that changes everything. The shape doesn't matter. It doesn't matter if what the junction angle is. Whenever you have the junction angle, you'll see the following thing. If the flow speed is too low, here Reynolds number just over 200, all the bubbles that come in leave. So nothing is trapped. You double the flow speed, and the bubbles get trapped. So what this is also telling you is there's a bifurcation in the flow. The feature of the flow changes above a critical Reynolds number. And it turns out that Reynolds number is related to the angle. Okay? And so we've mapped all of this out. And so if you were interested in this, we've now made a little diagram that says, you tell me the Reynolds number, you tell me the junction angle, and there's a region where you will trap bubbles. Below the lower boundary, the flow isn't strong enough, so you don't trap. In this region, you trap. If you have a given shape and the Reynolds number is too large, the flow is turbulent and everything gets blown out of there. Okay? But there's always a region of several hundred in Reynolds number where you trap. The last 5 minutes of the talk, I'm going to tell you where this solid line comes from. It basically comes from an argument for how this happens, and we can rationalize using a single-phase flow idea the bottom boundary. Okay? Okay, so flow in a channel. So your elbow, if you want to explain to your friend what flow in an elbow looks like and how it's different than flow in a straight pipe, the fluid mechanical picture goes back to a man named Dean 80 years ago, so it's called Dean flow. And you know what Poisson flow in a pipe is like. Those are the solid lines coming out at you, telling you lines of constant velocity. But when you go around a curve at modest Reynolds numbers, the flow has a secondary flow structure. That's the curve line, so I'm going to try and do it here. If I'm following the fluid and I'm trying to turn a corner and I'm following Mr. Dean, if this is the inside of the corner, labeled I, and over here is the outside of the corner, labeled O, the flow, I have to get this right, goes from the inside outside. So as I go around the corner, I'm having a senior moment again. Yeah, as I go around the corner, the flow is doing this. Two vortices get formed, like that. Okay, that's what this is trying to show. So this secondary flow is telling you it's not a closed vortex, but an open vortex. There's a flow coming out at you. If I now show you this movie again, now look at this movie again, realizing that the flow is three-dimensional, and there are two vortices sitting in the flow on either side where you turn the corner, you'll start to see there's a line of bubbles, there's another line behind it. They're marking two vortices in the flow. And if you're a turbulence flow person, you know you find vortices by putting bubbles in a flow. Here, what's unusual is the bubbles don't leave, they get trapped. So it's not simply, like a turbulent flow where I just put bubbles in and they find vortices. So in this flow, there are vortices formed naturally by the flow, and those bubbles tend to accumulate and then trap bubbles. Okay, so that's kind of the Dean flow idea. Okay, so in the fluid mechanics literature, I was aware of this at the time, though I didn't know what it meant. There's a term called vortex breakdown. It's normally only studied in the fluid mechanics problem by people who take a straight pipe with fast flow along the pipe, and they add a lot of swirl to the pipe. And when you add a lot of swirl to the pipe, above a critical swirl speed, the fluid comes in if you put a little dye. There's a burst that forms a bubble-like structure of ink, and then the ink slowly leaks out. And this cavity is called the vortex breakdown bubble. And it's always associated with, I'm going to go out on a limb again, these kind of artificial laboratory scale flows where you have a straight pipe with artificially induced swirl, and above a critical speed, you get a vortex breakdown bubble. I'm now going to claim that in the standard T junction, in every engineering system, this is possible, even though the flow is changing direction, because vortices get formed. There's a little sketch from my colleague at Princeton Phil Holmes about what this might look like, that inside the flow, there's a trapped cavity of liquid. Okay, this is what might be weird to you if you're ever thought about flow. I'm going to claim that inside an ordinary flow, you can trap pockets of fluid that just circulate. Here's a numerical simulation that has the idea, and then I'm going to show you an experiment, and then I get to almost stop. So we got lucky. I had a visitor. There was a visitor in another department, Stefan Rodel, who was an expert on numerical methods. And one day he walked into our lab with a numerical simulation, three-dimensional numerical simulation, that suggested what was going on. So here's streamlines shown in red. Yellow means regions of high vorticity, high swirl, if you like. There's streamlines that leave. They come in and leave. Above a critical Reynolds number, here close to 600 for a 90-degree bend, there's streamlines that are going in a circle, going on a tube, if you like. They hit a stagnation point and come running back down the middle. There's closed streamlines inside a fluid flow. Jean-Pierre, you ever seen something like that? Okay. So this is what the numerical simulation shows. We've shown it with 3 different numerical simulations, simply because one year my proposal got turned down by people saying it was all numerical error, even though I had an experiment in the thing. So this feature, I'm going to argue, is a vortex breakdown-like structure, just like this laboratory experiment I showed you before. A bubble, a pocket of fluid, where all the fluid's going past it, and it's just circulating inside. To give you an idea that this is real, here's an experiment with bubbles. There's a large bubble that's formed from coalescence of many bubbles. It's stuck right at the middle. You can see the top of the pipe at the bottom. This bubble is stable as stable can be. This isn't an unstable point. It's a very stable point. If you follow some of these bubbles, they come in, they spin out around this thing, and then they come back down the middle, just like the numerical simulation shows. So here the bubbles are acting like tracers on the flow. So what this flow does, what this three-dimensional flow at this T junction does, which I'm going to claim is very common over ranges of hundreds of Reynolds numbers, is the change in direction creates this vortex. The vortex above a critical Reynolds number has this so-called breakdown phenomena. It has a stagnation point in the flow that causes a trapped pocket of fluid. And so the bubbles simply can't leave. They get trapped by the vortex, and above a critical speed, or below a critical speed, they get trapped. Okay, that's kind of what this has to do with lots of things. We've done lots of numerical simulations. That's telling me I'm almost done. Maybe in the interest of time, I'd rather answer questions. So there are lots of questions you could ask about this. We've been trying to ask many of them, although the geometry means it's hard to do anything with pencil and paper. But you can ask, given this, how might you clean systems that have these trapped pocket of fluids above a critical Reynolds number? We've said certain things about, we've gone back to study the Dean flow problem. My colleague Clancy Rowley likes the term bifurcation at a bifurcation. The flow has a bifurcation. The T junction looks like a bifurcation. Okay, so here's your quiz. Okay, given what I just told you, suppose someone says to you, clean ink out of a pipe. The pipe just happens to have a T shape. Please tell me how long it's going to take, and I'm going to tell you how fast the flow speed is. Okay? So all we're going to do, this is ink in a pipe. We're going to turn on the flow, and we're going to ask, how long does it take to clean the pipe? And you're going to say, can't be long, right? So there's a long lead time here. So you turn on the fresh water. The fresh water will come in. You'll start to see it rapidly clean the left part of the pipe. It rapidly cleans it, except there are these two vortex breakdown bubbles which only slowly leak ink because there are these trapped pockets of fluid inside the flow when the Reynolds number is in this critical range. So anyways, that's my little short story about the T shape at finite Reynolds numbers. So those are the three stories I told you. I tried to tell you a little fluid mechanics story. I've only picked very simple geometries, but that's why I think if there is relevance to the work, it might be because these geometries are fairly simple. They're not complicated. I showed you the upstream walking. I showed you the biofilm streamer, and I showed you the trapped bubbles. And with that, I've used 50 minutes, so I will stop and answer questions.
Thank you for speaker first.
See, you were so stunned you didn't know what to do. Well, I hope it was interesting. I mean, those are my three. My group does other things, but those are my three stories for today.
Okay, yeah, Michael already have a question. Yeah.
Does the rheology change with time when this? Does the rheology change with time when this polymer falls?
So that's a great question. Exactly what the rheology is. There are now a number of groups around the country and around the U.S. at least in engineering departments, and actually one in Alberta, I think, that are now trying to collect enough biofilm that you can measure its rheology. There are early studies of this. Does it change in time? That I don't know. What is probably true is, for any given rheology, as the biofilm gets thicker, it's effectively softer. It's just easier to shear. But its rheology, I'm going to guess, is probably not a strong function of time. But as it gets thicker, the whole system, as you know, from just shearing thin films, is effectively softer.
Okay, yeah.
Yes, with the experiment that you did with the biofilm streamers,
Yep.
if you did those experiments with dead bacteria, would you still get the streamers?
That's a great question. So the question is, what about dead bacteria? So the way we did that, we used colloids. So we did one experiment one day where we took the bacteria. You spin the system down to remove the bacteria, but you still have the polymeric material it produces. You add colloids and you do the experiment. And we do see structures a little like this. The bacteria, I think, is simply helps you see it. What's really taking place is the polymeric material that's sticking to the surfaces. And if you ask, have you ever done a completely artificial experiment? No, we haven't, but we've wanted to. And you could also ask, well, maybe I should let you ask. But there are blood flow issues that also you could think about in this context. We tried some. One summer we bought blood from a blood bank and tried to do similar kinds of experiments, because whenever I give this talk in a bioengineering audience, they often ask this. All that happened, Princeton doesn't have a medical school, so this might have been important. We could never get fresh blood. The lab stunk, and my group nearly shot me. And it's also hard to see into it, so we didn't really draw any conclusion from it. But certainly when you read about the blood flow problem, it has many similar words associated with it. So I think it's a good thing to do, yeah.
Just a purely technical question. When you had, for your first part of the talk, when you had the leaf and you had the regular movements, what was the source of the fluid flow? Were you pumping fluid through the leaf, or was that? Yeah,
so it was just attached to a weak pump.
Okay,
Yeah.
so
Yeah.
you were pumping
Yeah.
through it. Awesome. Okay. Thank you.
Nessa? Yeah.
So you spoke a lot about what happens after streamers form. I've been clear about why they form. You mentioned a stagnation point.
So exactly why they form in the first place.
Yeah, because if it's a stagnation point, it doesn't. You need the bacteria to be positively buoyant for them to kind of migrate there.
Okay, so it's a great question. I'm going to suggest an answer, but I'm not going to claim that I understand this, exactly how it happens. But I have one slide that tried to suggest how it happens, which is here. There are no internal stagnation points here. The only stagnation points in this flow are the surface. The whole surface you can think about as a zero velocity boundary condition. It's as if this is a pipe, but on the surface is a soft material. So this movie at least suggests how things get into the flow. And that is, you see in the neighborhood of the corner, you will see a filament form. Okay. Now, where we think it comes from is this picture of a single phase flow. We did lots of numerical simulations and some asymptotics. And I tried to give this sketch of, well, you know pipe flow. Anything that causes flow out of the plane is called a secondary flow. At low Reynolds numbers, okay, so at high Reynolds numbers, I drew the picture of Dean. I did this. That's not true at low Reynolds numbers. At low Reynolds numbers, there are these colored red and blue that are telling you the vertical component of velocity. They are opposite each other around the corner. That's, at low Reynolds numbers, the Navier-Stokes equations are linear, and there's a mathematical feature associated with that, which is the secondary flow has to change sign about a symmetry plane. So upstream of the corner, the secondary flow in the low Reynolds number case, upstream of the corner is going away from the middle. Downstream of the corner, it's going towards the middle. So the suggestion we made is over a long time, if I have a soft material, maybe I'm accumulating material near the middle. And then, due to some instability, you can extrude, possibly extrude a filament, and that filament then will catch material. Okay. Now, why is fluid mechanics important in that? If I give you a flow with curved streamlines, and I put a very thin filament in it, you might argue, shouldn't it just follow the streamline? Can you help me? Couldn't it just follow the streamlines around the corner, right? Maybe it just follows the streamlines. It's a little filament. It's a polymer. But follow the streamlines around the corner. Okay, but now you ask, if the filament is under tension, like it's being held at one end, then you have a force along the filament and a force along the filament, and the sum of those two vectors is across streamlines. So you can't have a filament that simply follows streamlines. If it's under tension, it has to cross streamlines, and that's what you see here. It has to cross the streamlines. That's a feature of the elasticity of the problem. And so we have tried to write a couple papers where we tried to combine fluid mechanics and elasticity, and at least they're suggestive that this is not an impractical way to think about this problem. So it's a fluid elastic problem if you want to understand the dynamics. But your question, how does it form in the first place? I'm stuck with this. There's some fluid mechanical instability that's taking a soft material and putting it in the third dimension. And I've tried I'm not so good with pencil and paper anymore. I've tried to cast an instability problem. I've never made it work. The one I know in the literature is always on a plane boundary. There's a whole literature on that. Very nice. But here what's important, I think, is the corner, because we only see these at corners. And I don't know how to make that work. Michael.
Will the extensional property of the hydrogel as the molecular increase that you have these?
Possibly. No, it's a good point. Possibly. There's certainly some extension happening. That's how this thing creates this three-dimensional structure. And my point is, of course, that once you make it three-dimensional, once you put something in the flow, then the whole character of the flow changes. I showed you evidence for that. So it's important. But I don't know how to answer your question. I think it's the one question we've kind of qualitatively said, we think the following happens, here's the evidence. But how to describe it, mechanically, write an equation, don't know.
So Andrew, you have one other question? No. Okay, sure.
Yeah.
We'll probably take a few more questions, then we have to stop. Thank you.
Well, you see across the stream, because the streamline should just be following the geometry.
I was just thinking about the assumption that you showed earlier with no slipping.
Yeah.
Which I guess it's not really accurate in this case.
So your statement is the no slip condition.
In the center?
Yeah.
I think he means the simplification of the single filament down a straight tube.
Okay, so there I think, the way I would state that is, I think I see what you're saying, because you're saying I have this thing moving in the flow, right? The way I think I want to say it is, once this is formed and it's roughly stationary, then the velocity of the fluid is so much faster than any small slip that will happen because of things sliding along the filament, is such that the no slip condition isn't bad in this configuration. And if you ask, how does this form? If I put this filament in the flow and it tries to follow the flow, that's a no slip condition. So I think the no slip condition is a good first approximation here. Those of you who know me know in microfluidics, I think the no slip condition is always the right first approximation for everything, for all practical purposes. But I don't think they're inconsistent.
Okay. Yeah, David, you can go.
So regarding the T junction,
Can you speak a little louder?
the T junction vortex trap.
Wasn't that cool? Did you like that?
Cool. Okay, yeah, so the third project that you mentioned.
Yeah.
So I remember some of the compressed air, cold air gun that is used in industry for cooling CNC
Yeah.
machines. There is a thermal effect. It's a similar, but I'm sure they have a higher Reynolds number. Have you think about using, is there any thermal effect?
Wait a minute. The thermal effect associated with separating, there's a way you separate gases by vortex tubes. Is that what you mean? Okay, I'm probably going to get this wrong. There is an effect that goes back to Rankine or something that's called the vortex tube, where you get in a pipe, where you get a temperature difference associated with the fact that you have a swirl and you have gas molecules of different molecular weight, something like that? Is that the effect you're referring to?
No, that just
No.
doesn't need any modification.
Okay, so well then let me answer the question this way.
Because of the swirl, there is a significant pressure difference that has a significant temperature difference.
Yeah, okay. So it's true here that what is important is the fact that when you go around a corner, you get a vortex. And what's important to me is that you get the secondary flow, this what I'll call axial vorticity, the fluid's going downstream like that. That's important. Why? Because if you have, in this case, water of a certain density and I put in a low density object, it wants to go towards the center of the vortex, not out of the vortex. That's why you trap low density objects. We also trap, by the way, rigid spheres that were hollow. Anything that's roughly lower than some critical density, you trap. So that's certainly important. That doesn't explain everything, because they should be leaving the flow by your explanation, whereas what I showed is they get trapped, and they get trapped because above a critical swirl, the flow ends up with this funny internal stagnation point and starts to circulate. So part of what you say is certainly true. Now, if you're saying, will this happen in airflow in a pipe, that I don't know. Well, that's not true. I do know, because I'm claiming it's a single phase flow effect, and if you tell me the Reynolds number, there's a critical Reynolds number where you get this trapped pocket of fluid. That's a fluid mechanical effect. Whether or not it's have anything to do with what you see in heating and cooling, I don't know.
Yeah, we'll take one last question. Yeah, then we'll finish.
Last question. So, your main parameter was the Reynolds number, but the Reynolds number is a combination of different parameters.
You mean for the third part of the talk?
Yeah.
Yeah.
Okay, so what you are changing exactly there? Is it the density or the fluid velocity?
So in the experiment, it's easiest to change the flow. In the numerics, we just change the Reynolds number. So in the fluid mechanics problem, if it reduces to the Reynolds number, you can change any of them. The only parameter that matters is Reynolds number.
But then would you expect the same phenomena or results happening on a large diameter pipeline?
If the Reynolds number is the same, yes.
The same for
Yeah.
blood vessel?
If it's just the Reynolds number. You tell me the Reynolds number, then it doesn't matter what the size of the system is. That's the beauty of fluid mechanics.
Okay, yeah, due to the time limit, we still have the reception afterwards. We're going to stop here for the results and discussion, and it's really exciting. It's the first time I saw during this talk, we had this dynamic discussion. Let's thank our speaker once